Are we good to go? Should we start? Yes. So it is my great pleasure to welcome you to the Genome Application Symposium. Um, I am very excited for these 2 days of discussion about applications on biodiversity genomics. My name is José Melo-Ferreira. I'm a researcher at CBU Biopolis at the University of Porto and professor at the Faculty of Sciences at the University of Porto. I'll pass the word to Elena, who is also involved in the organization of this genome applications work package in Biodiversity Genomics Europe.
Thank you, José. I would just also like to say hello to everybody and welcome to the Genome Symposium. I'm Elena Buzan and I'm coming from University of Primorska, Slovenia, and I'm also a professor in molecular ecology. And I hope that we will find we will learn quite a lot of new insights today and tomorrow regarding application in biodiversity genomics. So thank you.
Thank you, Elena. So this is a very brief welcome session. I prepared some slides just to provide some context of why are we organizing this symposium. And I will now share my screen. Can you see my screen? Yes, and I'll put it in presentation mode now. All good? Okay, so again, welcome to the Genome Application Symposium. So in this first very brief opening session, I would just want to welcome everyone and provide a little bit of context of why are we organizing this symposium and describe very briefly the program that we have for today and tomorrow. So the technological and analytical progress that came with genomics has revolutionized the way that we are able to study biodiversity. And the European Reference Genome Atlas came in resulting from the recognition that we are at a time where we are actually able to generate reference genomes for all biodiversity. And this can be a very important basis to develop further genomic applications that can actually be used in conservation practice, but also exploring other biodiversity aspects. With ERGA and this network of researchers with this common goal, several different papers have been came out recently, and these are just some of these papers. And these essentially have put forward and proposed and recognized the importance of reference genomes for conservation genomics, which we can see here in the middle. Also explores how genomics can help biodiversity conservation, so the potential of the diverse array of genomic analysis in the conservation of biodiversity. And then I put here just a third that there's a preprint available, but has been recently submitted to molecular ecology, where we discuss how harmonizing genomic research is very important to meet stakeholder needs for conservation management. And so this symposium comes within the Biodiversity Genomics Europe project or consortium, which embraced the challenge of exploring and developing and demonstrating the application of genetics for several matters of biodiversity application. And this Biodiversity Genomics Europe project puts together two European networks, IBOL Europe, focused on the development of barcoding-based tools for high-scale, large-scale monitoring, and ERGA, which I mentioned before, which puts its emphasis on the generation of reference genome and associated genomic analysis for biodiversity research and application. And so within BGE, one of the work packages that is organizing this symposium is called Genome Applications. And I think these two words, demonstrate and engage, summarize very well the objectives of this work package. So the aim is to demonstrate how genomic data anchored on reference genomes can actually be used to develop applications that solve real-world biodiversity problems. And then this can only be done, of course, engaging all the actors in this stream of events that are required to apply genomic analysis, from scientists to the end users of these applications, the stakeholders. And within this work package, within Biodiversity Genomics Europe, this work package is organized in two types of tasks. One is developing case studies. So these are research and development projects that aim to demonstrate how genomic data anchored on reference genomes can be used to address real-world problems on conservation, bioeconomy, and disease. And so we are developing a series of case studies focused on a variety of different models to address this challenge. We have then a second set of tasks, this Biogenome App Network and Genomics for Society, that provide essentially support to the development of the case studies, promoting networking, not only within BGE, but also looking beyond BGE to the broader European communities, to exchange ideas and develop conversations that can be used to promote the development of these genomic applications. And then the Genomics for Society occupies itself from the last part of this stream, so the connection between this applied research and stakeholders to promote and maximize the application potential of the applications that are being developed. And so this Genome Application Symposium comes to respond to this challenge, to put people discussing several different aspects in biodiversity genomics research. It is more focused on the upstream part of developing genomic applications, but we also have tomorrow morning an emphasis on this downstream part of the stakeholder engagement and communication. Actually, we will discuss how it is important to engage stakeholders from the start in the development of the research projects and the aims of the application development. So I won't go into detail into all this, but we will have several different, and you've certainly seen the program. We will talk about adaptation, we will talk about unconventional species, some techniques like low coverage techniques, how it can be used and maximized in genomic application, temporal genomic analysis, species of economic value, plant genome applications, and we will have an emphasis also in the genomics at the Mediterranean basin. And in addition to this, we will present some preliminary results of a gap analysis that we are developing within this work package in Biodiversity Genomics Europe, focus on essentially the overarching question of what can we do best to improve the applicability of genomic research. And I will just end this welcome session by acknowledging and thanking the people that have been involved in the organization of this symposium, João Pimenta and Christian de Gutri, which are not present here, but also Luisa Marins and Chiara Bortoluzzi, João Marques, that have been really important to organize this event. And of course, I would like to give my warm thank you to all the chairs that have embraced this opportunity, but also responsibility of dynamizing these different sessions, and I'm really appreciative of your enthusiasm in this to participate in this event. So I won't take more of your time. So this is the end of the welcome. In approximately 4 minutes, we will have our second session, which is the first session for discussion, where we will discuss challenges in conservation genomics from analysis to essential biological variables, chaired by Ivana Bui and Camila Mazzoni. And I will now stop sharing the screen and ask for your patience for 3 minutes to start our next session. Thank you all for coming.